So, hello, good evening, everyone. Um, we have just a few minutes to the beginning of today's lecture. Don't forget, today this is another opportunity to actually meet with you on GLT 302. GLT 302. By my watch here, I have like 2 minutes to the commencement, the real commencement of this lecture. Don't forget that you are to make comments that are very, very relevant to the video. Make comments, subscribe, like, share. Don't forget, and you are to comment more than 3, 4 times before your comment will be visible. So kindly do that, and you are to, for some of you that are just subscribing today, you might not be able to make comments because of one reason or another, and it is a kind of setup on YouTube. So while we wait for others to join, permit me to really tell you that your exam is forthcoming, very, very close and very much around. Don't forget to visit my store, my online store, that is omega.seller.com. Yeah. Pick any ebook that is relevant to your area of specialization. So don't forget, this is another opportunity for us again. So without wasting our time, that's really wasting our time. I am going to go straight to the topic of today, and that is general instrumentation, which is actually meant for microbiology and environmental students. This lecture is specifically tailored to environmental biology as well as microbiology students. Any area of microbiology will actually require that, and that's the reason why um, this lecture is very, very important for you. So without wasting our time, I want to believe that others will be joining us. I can see some people are already making comments. Some people are already available online. So um, the course is general instrumentation. General instrumentation. And laboratory instrumentation are essential. They are essential tools. So it's just like every profession will actually need tools or instruments to actually work with. Our forefathers that were peasant farmers, no farmer can go without any tool to be able to do farming. The same thing, if you are going to be a microbiologist, if you are going to be any aspect of science, you actually need what? You need tools, you need instruments, you need something that is going to assist you. If not because we have the tools, so many people will have been lamenting, especially when it comes to the issue of making some headway. So, laboratory instruments are essential tools that we use in any aspect of in any aspect of biology, any aspect. That is, why do we need the tools? Because in science, in biology, we depend on observation, and our observation must not just be ordinary observation. If somebody says, "I can see you," that is unscientific. If without anything to document the intensity of the light that is entering. So that is why we actually need observation, measurement, analysis, and control of biological and environmental, I mean, samples. So we need that. Without wasting our time so that it won't be so repetitive. In the early part of this course, I've actually taught you about a basic laboratory instrument such as the microscope. So I told you what a microscope is. It's a magnifying object. And before the advent of microscope, it actually started from somewhere. And what are the different types of a microscope? We have light microscope, which uses. So if you want to write anything for us, even in the exam, I want you to actually focus on the types, uses, and application. So that is what I want you to focus on. So light microscope, what is it being used for? It is used, it uses the mechanism, it uses visible light and glass lenses. That is how it appears. And magnification, it can magnify object up to 1,000 times. That is what we talk about oil immersion, oil immersion lens. So in those days, when we were talking about the magnification of, if you are using hand lens, it's quite different from when we are using objective. When you pick up a microscope, that microscope can either be monocular, that is, one is having just one eye, that is only one point that you can use to observe, and that's what they call the objective lens, only one. So the one that is very close to your eye, they call it eyepiece, and the one that is very close to the object, they call it objective lens. So when you now combine, the objective lens is going to have a specific magnifying, so it may be just times 20 of the object. Is going to magnify it times 20. So it is the image of the objective lens that the eyepiece will pick and is going to send it to your eye, and that's why they call it eyepiece. So it uses visible light. It depends on light. The power, what powers it is what? Lens, is light. And for some of us that are, let me just say all of us, we remember what we learned in physics about lenses, light. So we know that. So what is the application of that? It is used for observing bacteria, fungi, and protozoa. And we can also make use of it to be able to see some dyes, how this object appears under the what? Under light, I mean under stains. So the second one is phase contrast microscope. I won't waste our time because of on this, because we have actually gone to pick the, I think I have done this for you in first second lecture, and this one is also going to be available in my seller store very soon. Maybe at the end of this lecture it's going to be available, so you can pick it up from there later on. But this one is just going to be direct to the is more of an exam focus than anything. So, and don't forget that during the examination, this is the way I want you to write. This is not history. This is biology, and it is the science of precision. The science of precision and accuracy. Just go straight to the point and tell me what each of these is being used for. Okay. So, and I'm going to make sure that the amount you are going to pay for this is not going to be more. It won't be more than 1,000 yeah, thereabout. So please, you can pick it up at the end of this lecture. Okay. So we, apart from that, the second, apart from microscope, the second biological instrument that as a biologist you must be familiar with are the autoclaves. Autoclave under autoclave operate under what? The sterilization, under high pressure. That is where they operate. So they operate under that because they believe that there are some microorganisms, especially some bacteria that are thermophilic, especially that even if you heat, for example, now if you say, okay, oh, you want to sterilize, you want to remove life, aseptic, you want to prepare your sample in an aseptic condition, that is in a condition that is free of contaminants or free of contamination. That's what we call abscesses. But now, under normal circumstance, as a biologist, as a microbiologist, you should be able to make sure that you do not introduce contaminants onto your samples. Don't introduce contaminants onto your samples. That means your sample must be very, very pure, very free of external life, that you will not be saying that you are working with microorganism. What you can see is microorganism, and at the end of the day, you are not you have introduced, for example, now if you are working with food sample, how will you, if you do not handle it very well, if you do not uh, use the principle of sterilization, if you do not use it to remove life from it, at the end of the day, you are the one that will now mishandle it. So instead of you to report, you'll be reporting something that is not even true. So that means your result is going to be false. So an autoclave operates with is a sterilization instrument that operates under steam pressure. So they use high pressure to be able to do what? To sterilize, to be able to make sure to destroy microorganisms. So, and that's the same principle that our pressure pots or pressure cooker actually work with. So, and that's why that in some local laboratory, what you see, instead of them to buy autoclave, what they will be using is a pressure cooker. So it operates under high pressure. They depend on high pressure to be able to make sure that they remove microorganism from it. And what is the principle? The principle behind that is that when you maintain high pressure even at 121 degrees centigrade, don't forget that is the boiling the boiling point of water is 100. So that means, and at that 100, there are some microorganisms that they are just starting their own life. But if that water is put under high pressure, that pressure can even work even at 121 degrees centigrade. And at that level, at that temperature, most of microorganisms will not be able to survive it. And that's the principle autoclave actually work. So under pressure, it kills bacteria, kill the spores of fungi, as well as some viruses. So how do we make use of it? You use it sterilization of culture media. So by now, some of us, okay, so some of you might have known, or you might have been taught what culture media are. Culture media are just like the food that you give to microorganism in order to be able to maintain their what? To maintain them in the laboratory, their culture media. So just like when you want to domesticate an let me say an animal, a wild animal, that means if you are bringing that animal to your house, you should be able to maintain that animal so that the animal will continue to live in the environment, that it will still look as if they are still in the jungle. So the food of microorganisms are called the culture media. That is, this culture media is going to supply them with most of the most important nutrients that they will need. So they use it in sterilization of culture media, We also use it in sterilization of glassware and surgical instruments. So not only our media. So at a point in time, you just put all your instruments, the instrument in the in the biology in the lab, your glassware, your conical flask, your scalpel, all this one. There are times that you have to put them, subject them under high pressure so that it's going to remove microorganisms from them. Then the contamination of laboratory waste under normal circumstance. If you want to discard any laboratory sample or any laboratory waste under normal circumstance, before you be able to release it into the environment, you must subject it into what? Into sterilization procedure. That is, you autoclave it before you be able to. So under normal circumstance, I've seen some people that they will just work in the laboratory, the culture media. So the one that they actually need to discard, they, instead of them to discard it, uh, autoclave it before discarding, they will just go and put it in the environment. So by the time they put it in the environment, they discard it in the open. Don't forget, they have polluted the environment. So that is that is autoclave. Now incubators. An incubator is used to maintain controlled temperature condition, so for microbial growth. So autoclave is sterilization of equipment, sterilization of culture media, sterilization of waste under high pressure in order to be able to remove microorganism from your samples. So that is that. Then incubators. And incubators is an instrument or is a device that is used to be able to maintain temperature. So for example, if you say microorganism that is from human sample now, under normal circumstance, they have to stay, you have to maintain them in the temperature that is commensurate with what the temperature of human body that is going to be. So if you want to maintain them at that temperature, you need an incubator. So under normal circumstance, it function provide optimal temperature, just like I said, like room temperature. It's not every time that when you say the room temperature is 37 degrees centigrade. There are so many times that sometimes it may be low and sometimes it may be high, depending on it. So if microorganism that exist in that condition, when the temperature cannot be controlled, you just see that that is not proper for them. So you see them behaving. And under normal circumstance, depending on the temperature in which you put them, okay, that will actually tell you whether they will be able to grow well or they won't be able to grow well. Sometimes they might even grow well and they will become so wide. They become more virulent. It's just like under normal circumstance, when you tame your dog, when you teach your dog, you tame your dog, and you are not giving your dog um enough support, you just discover that what's going to happen is that everybody will be running away. In fact, even you yourself might not even be safe. That is that. Then another thing that you is very common in the biology lab that you need to know is laminar flow hood. Laminar flow hood. If you check the replica of laminar flow hood is what we have inside the inoculating room in the biology lab. So although it was locally fabricated, but what that one was actually meant for is to be able to provide a sterile working environment. So sterile working environment. Under normal circumstance, if you are working in the open, if you are working in on the you are making use of the table as your working benches, you will not and you might not be able to maintain that environment as very, very safe. So in order to be able to make sure that the condition which you are is a very sterile condition, you make use of a laminar flow. Laminar flow means that when it has to be, it's going to be operating at specific temperature, at specific temperature, pressure, and what have you, which is going to help you in order to be able to maintain the integrity of your culture. So the main objective of a biologist is to make sure that you keep your what? Your sample free from contaminants, free from contamination. And that's one of the reasons why you, as a biologist, what you make use of is you make use of the PPE, what you call personal protective equipment, like you make use of your nose mask, you put on your lab coat, and so on and so forth. So laminar flow. Then another one is a biosafety cabinet. Biosafety cabinet, depending on the organism, the virulence of the organism you are actually working with. There are some organisms that uh, they exist, they are maintained at BS BSL 1, BSL 2, and biosafety cabinets, like that. So you need to maintain that. So protect the user. It's going to protect you as the user. It's going to protect the sample as well as the environment. It is used in handling pathogenic organisms, then working with hazardous biological materials. Then you also need to be familiar with what is called the centrifuge. A centrifuge is used to separate substances based on their density by spinning them at very high speed. So the principle is that centrifugal force, so for some of us in physics, you know what centrifugal force are. So centrifugal force cause heavier particle to separate. Those ones that are heavy, they will separate below, and those ones that are lighter or that are light, you see them, they float. So that's why the first thing is that when you collect blood sample, so under normal circumstance, if you really want to study the components of cells, I mean what blood sample, you will just discover that you will subject them to higher centrifugal force so that all the components of the cell, they will now be able to separate out based on their weight and based on their what is called the density. So there are three different types of what centrifuge. Number one, we have bench centrifuge, we have micro centrifuge, and we have ultra centrifuge, depending on the sample and depending on the laboratory in which you are actually working with. If you are in molecular biology lab, you are going to need more of a make use of microcentrifuge as well as a ultra centrifuge. So which uses separation of cell from culture. You can use it to separation, I mean for separation of cell from culture media. Okay. So isolation of cellular components, just like in blood samples, then clarification of different samples. So it is due. So the part B, which has to do with analytical laboratory instruments. So there are, apart from the general laboratory, there is a different laboratory which is being used by the three major science courses, that is physics, chemistry, biology. So in between, where you have chemistry lab, in between chemistry and biology lab, if you look at our new lab, you just see that the instrumentation room is between microbiology lab and between the chemistry lab, because it has to do of more of analytical instrument. And all these instruments, they are they are more expensive and they are very sensitive. So the first one we are going to look at is spectrophotometry. Spectrophotometry, and that is what we call spectrophotometer. So the basic principle of this is that absorbance is proportional to the concentration. So, and that's the principle they use in operating on. Okay. So the next thing is so the different types of spectrophotometer. Number one, we have UV visible spectrophotometer. We have infrared IR, and we have fluorescent spectrophotometer. So unfortunately, we don't have most of these in around us here. So where you can easily see that one is when you go to higher laboratory. Okay, you will see. Then the application, it is used for measuring microbial growth. If you want to measure, to really study microbial growth, you make use of spectrophotometry, abi. Okay, estimating proteins and nucleic acid concentration. Don't forget, I told you the analytical is more, you find them in you make use of them in molecular biology. So you use it in digestion of protein, enzyme assay, different types of enzyme assay, and so on and so forth like that. And the other one is chromatography. That is what we call the chromatogram. Chromatography is a technique used to separate components of a mixture. Components of a mixture. So that is the general principle is that separation occur due to differential movement between stationary phase and the mobile phase. So and there are four different types of chromatography or chromatograph. We have thin layer, which is called the TLC. The second one is IPLC, gas chromatography, and the ion exchange chromatography. So the next instrument we will also have to look at is electrophoresis. We actually need this in molecular laboratory. We can't do without that because that is what we use in digestion of, also as to be able to visualize DNA molecular samples. Visualize immediately after you must have run your PCR. How you can, how will you be able to see it? How will you be able to uh, see what has actually gone on in terms of sedimentation? You visualization. What you see is what you make use of is electrophoresis. So different types. We have agarose gel electrophoresis, page, that is polyacrylamide gel electrophoresis, and we have a two-dimensional electrophoresis as well. So uses, it is used for DNA, RNA, and protein analysis. That is molecular work. Then molecular biology research, it is also used for that. So the next thing is that the next instrument is a flow cytometry. Flow cytometry analyze cells as they pass through a laser beam. So we will make use of this. We will know more about this when you go higher. And when at which point are you going to be talking about? And that's when we'll be talking about cell counting, immune and microbial studies, and so on and so forth. So one thing I want you to know is that both basic and analytical laboratory instruments play a very crucial role in biological science, not only in microbiology, also in biochemistry, anything that has to do with uh, anything with biology. We cannot just joke with them because it is what we use in to record our observation, to document our observation, to be able to visualize, to be able to use as a kind of tool for this. So because it, and when you understand the basic principles of all these instruments, honestly, heaven is just your beginning. So because understanding and handling of this instrument ensure accurate results, safety, that is, you are also very safe, and it is going to be reliable scientifically. Okay, going to be reliable scientifically. So just like I said, that these are the two major things that we actually need to do in this. Okay, um, don't forget what I said, that we started with the basic. What are the basic instruments that we use in the laboratory? Microscope, after microscope, centrifuge, autoclave, incubators, laminar flow hood. You need to know all these. Oh, laminar flow hood, biosafety cabinets. We have BSL, BSL 1, BSL 2, BSL 3. For example, if you are dealing with organism like Mycobacterium tuberculosis now, that is at BSL 3, very high, because they are they are in terms of they are in fact they are very very virulent. They are virulent, so you can't just joke with that. You need to be extra and extra careful. So it's not just anyhow. When you go to TB lab or this, they don't allow anybody, just anybody anyhow. And the way it is even being constructed, it is a you meet you mean you come across a lot of partition where, okay, there's a place you have to, when you are coming in, you have to change dress in laboratory way, so and so on and so forth. Then apart from that, that cabinet, you see it in a kind of cabinet to the level that okay, maybe the next lecture I will be able to show you some of these pictures that you need to see to know them. So just like I said, it is what that you need to know, need to learn them. And don't forget that the examination is fast approaching. I actually want you to read more. Just like I said, this material is going to be available very very soon, and it's not going to be a token, so it's not going to be more than 1,000 naira. If but I might not even put it there, I don't know. I'll not even put it there, so that this, but don't forget that this is a instrumentation general instrumentation. That is, what are those instruments that you, as a microbiologist, as a biological scientist, what are these instruments that you need to actually learn about and you need to know about? So that's why they call it a general instrumentation. And I'm very sure that the other two lecturers, they will also do their own parts as well. So, without wasting our time, I want to appreciate you for your patience. Don't forget to comment, don't forget to like, and don't forget to also share. So, this is the end of the lecture. Okay, thank you. I'll see